Thank you.
Well, good evening, everybody. Boy, that sure is a tough act to follow. I first came to Stanford in 2004 as a clinical fellow in hematology, blood diseases. And right away, I was working in the hospital under the care of senior physician, training of senior physicians, in the care of patients with acute leukemia. And as I started learning about the way that we approach treating these patients, we give them chemotherapy for a week, but it's so intense that they need to be in the hospital for a full month. As I started learning about the clinical trials and the history of the therapy that we were giving, I was astounded to learn that it had been studied and implemented in the early 1970s. And that's when I was born. So I decided, wow, this disease, AML, acute myeloid leukemia, this is what I wanted to focus on in my research. And for my research, I joined the lab of Irv Weissman, the director of the Stanford Stem Cell Institute, and a pioneering scientist in the field of stem cell biology, blood, and leukemia. And we set out to try to understand what was really different between leukemia and leukemia stem cells on one side and normal blood and normal stem cells on the other. And in order to do that, I was able to take advantage of technologies and innovations that Stanford really pioneered. Our knowledge and understanding of blood stem cells from Irv, methodologies to take human patient samples of leukemia and be able to grow them in mice in a way that modeled the human condition. And then something called microarrays, a new technology at the time that allowed us to interrogate or understand the different types of genes that were involved in the disease without knowing which ones to study ahead of time. And it was these studies that then led us to the identification of a molecule or a gene called CD47. And that CD47 was much higher in its level on the leukemia stem cells than on the normal blood stem cells. Now, we weren't the first to come to CD47 in terms of its biology, and what was known was that it was an important regulator of aspects of the immune system. Now, we've learned a lot about our immune systems during the COVID pandemic, T cells and B cells, but there's another type of cell in the immune system, macrophages. Now, we like using these fancy terms in biology, medicine, and science, macrophage from the Greek, macro, big, phage, eater. These are the big eating cells of the immune system. And their normal job is to ingest, digest, and get rid of dead cells, debris, dying cells, as well as pathogens. Now, of course, they can't do this in an uncontrolled way. They have to have a system by which they're regulated. And CD47, it turns out, is one of the most important mechanisms by which these macrophage cells are regulated. CD47 on any cell will link with a different protein on the surface of the macrophages and thereby instruct them not to eat. CD47 is this don't eat me signal. And the leukemia cells, we postulated, had adopted this mechanism to evade being eaten. And that led to a very important hypothesis that had therapeutic implications. What if we could block that signal? Block the CD47 signal. That means that the macrophages would eat the leukemia cells. So we set out to test that in an experiment using an antibody, another type of a protein, but very specific for CD47. And we went to the lab, and I was working with a very talented medical student, Mark Chow, at the time. And we set out to do an experiment. We took those mice I described earlier, into which we had put human leukemia cells. And we took a sample of blood on day 1, and we treated them with the antibody, and took another sample of blood on the second day, and we're analyzing for the presence